Good morning, CP Biology students. This is Mrs. Groves. Welcome to Marking Period 4. We are at the end of our protein synthesis unit, and the last topic that's being covered is mutations. So at this point, you've done a couple assignments that involve mutations. You watched a review video on mutations. Now, the last thing I'm going to have you do is really see if you can perform mutations to DNA sequences and see how it affects the protein that gets assembled from that mutation. So today I'm just going to go through some of the big ideas of what you need to know in order to be able to solve these types of problems. So starting out, just what is a mutation? I know at this point we've covered this a bunch of times, but mutations are changes to DNA. So sometimes DNA can be altered by something in the environment, and this alteration in the sequence can lead to a change in the protein that is made from that DNA. And we call that gene expression, proteins that get created from the DNA genes. So this is a mutation. There's actually more than one type of mutation that can happen to genetic material, and we'll get to that a little bit later on in this presentation. Okay, so if we move on, mutations are changes to the original DNA sequence in the nucleus of eukaryotic cells. So if you change the DNA sequence, keep in mind what we've just been working on, where we take DNA and we copy it into RNA, and then we make a protein sequence out of that DNA. So if you think of changing the DNA sequence, that's going to change the mRNA that is transcribed from the DNA, and ultimately it can change the protein that gets created. Okay, and that can create things like genetic diseases. It can also create new traits. For example, we know that blue eye color is actually the result of a mutation that occurred in humans about 10,000 years ago. Okay, so before we look at some mutations, I just wanted to go back to what we covered before break. If you're looking at this picture of the cell, right, you've got DNA in the nucleus. That's the genes. They hold the instructions for all of the proteins that a cell can make. When a protein is being assembled, the DNA message is copied into mRNA. That's called transcription. The mRNA leaves the nucleus and goes to the cytoplasm where the ribosome is. And what the ribosome does is it takes the mRNA sequence, it reads it off in a series of 3 bases at a time. That's called a codon. And each codon gives us an amino acid that gets added to this growing protein chain. So this protein chain is the end result that we're going to be looking at. And you can see it here. Sometimes it's called a polypeptide. Okay, so just to recap before we get into mutations, I want you to just think for a second to yourself. If a mutation occurs during DNA replication, how will that affect a cell? So we know DNA gets copied before a cell divides. If you have a mistake in that copying, that mistake will then be passed on to the cells that are created from that original cell. And so the mutation can get duplicated again and again and again. Okay. Before we get into the type of mutations we're going to talk about today, which are mainly gene mutations, there are 2 main types of mutations that can occur. Gene mutations, which are sometimes called point mutations, versus chromosomal mutations. And we'll talk about these later when we get into genetics. Okay, chromosomes are large segments of DNA that have lots of genes on them. So if you have a missing or an extra chromosome, or a chromosome that's mutated, that's going to have a large effect on an organism since it contains a ton of extra genetic information. Okay, for what we're going to be working on, we're going to focus on gene mutations. So these are typically called point mutations because they occur at a single location in the DNA. It's usually 1 or 2 bases that are mutated in some way. Okay, so you should know some things that can cause mutations. You should know that mutations can occur randomly, but there can also be certain substances in the environment that can cause DNA to mutate, like radioactive material or UV radiation, certain chemicals and drugs, etc. Okay. So a mutation is any change in the sequence of nucleotides in DNA. We call that a gene mutation because it affects one single protein. And so at that single point, mutations there's a couple of different things that can happen. You can have a base that gets deleted, sorry about that, let me go back. You can have a base that gets added, You can have a base that gets substituted, meaning instead of an a T is put in place on accident, or you can have a base that's moved. We're really going to focus on substitution, addition, and deletion of bases in what we're doing. So if you look down here, let's see what type of mutation occurred in this example that I'm showing you. If this is the original DNA in the gene, you can see down here that now we've got a C, I'm sorry, that keeps switching, a C where there was a G. So did we add a base, did we delete a base, or did we change a base? In this case, we changed a base. So this would be called a substitution mutation. Guanine was substituted by cytosine. Okay, so let's just look at some of those mutations. Keep in mind that not all mutations can end up harming an organism. Specifically, mutations that happen in sex cells, in the sperm and egg that become an organism, those mutations are going to be more serious because they're going to be found in every cell of the organism's body. Some mutations are acquired in an organism's lifetime. So if you are exposed to X-rays, radiation, if you smoke cigarettes, etc., those are all called mutagens, or substances that can mutate DNA. So we said point mutations occur at a single point. In this case, I'm giving you an example of, instead of thinking about DNA in terms of A's, C's, T's, and G's, we know that the sequence of DNA is read off in 3 bases at a time, and we call those codons. So in this example, instead of using A, T's, C's, and G's, we're using 3-letter words to represent the mutation. So if we had the sentence, the fat cat ate the wee rat, these are each of our codons. Let's say we had C replaced with an H, a substitution. Notice how it still says the fat ate the wee rat. The only triplet that changed is the word cat to hat. Okay, so this is called a substitution reaction, or a substitution mutation, because you're only changing 1 letter and all the other triplets stay the same. Okay, so this can have a big effect on the protein that forms, but it also might not, because most of the other triplets are still the same. So substituting does not tend to have as big of an effect as when you insert or delete a new base. Okay, so if this was my sequence again, the fat cat ate the wee rat. Now look what happened when we add another letter in. So we inserted the letter A where this T is, and now the T is moved over, or I'm sorry, we deleted a letter. So we took away the T here. And so everything was moved 1 to the left. You get CAA instead of CAT. And now look at every 3-letter word that comes after that mutation. So we deleted a letter. I know I originally said inserted, my mistake. And now every triplet down the line is wrong. Okay, so we call this a frame shift mutation. Anytime you add a letter or remove a letter, you're changing that 3-letter sequence. That's called the reading frame at which the ribosome reads the sequence. So frame shifts mutations tend to change every amino acid down the line. They have a major impact on the organism. Okay, and you'll see that later on. So deletion again is where a letter gets removed. And insertion is where a letter gets added. Okay, so now what we're going to do is you need a piece of paper and a pen. You're going to do some practice problems, and those practice problems you're going to take a photo of once you're done and submit as your assignment for today. So if you need to pause for a second, hit pause, and you don't need that much paper, but you need a place to write out some practice work. The other thing you may need is found in your textbook. You need a codon wheel or a codon table in order to translate. So it's good to have this in front of you when you're doing these practice problems. So if you have your textbook available, page 367 has a codon wheel for you to use. This is a codon table. It works in a similar way. You find the first letter here, the second letter, and the third letter here, and where they all meet, that's the amino acid that codon specifies. Okay, so here are your practice problems for today. And I know if you want to hit pause for a second, you can look at it. What I want you to do is, you've got your original DNA sequence here. So copy this down, and I want you to transcribe it into mRNA and then tell me what the amino acid sequence would be. Okay, this part is without the mutation. And then we're going to look at two different changes to that original sequence. Okay, so if you want to pause it and just copy this down and complete this top part now, do that. I'm going to continue explaining. In the example below, now you see it's a mutation to this DNA sequence. So if I look here, I have GTG instead of GTA, and the rest of the sequence is the same. So what you want to do is transcribe and translate this mutated sequence and compare your protein molecules. Was the protein changed by the mutation? Would this mutation affect the organism? Why or why not? Okay, so in this case, I see a substitution happening. I don't see an addition or deletion. Rather, A is being swapped with G. And in the final example, down here, you still have a different mutation occurring. So, let me go back. Compare what happens in this example to your original DNA sequence. So now you've got, instead of GTG as the mutation, is GTT. Okay, I want you to transcribe and translate it again and look at whether or not this would have an effect on the organism. Okay, so I'm going to put up the answer key now. You are going to just check your work to see that it's correct. And here is what you should have gotten. So you should have seen that your first mutation gave you the same amino acid sequence, even though we changed A to G. Okay, this is called a silent mutation, okay, because the mutation causes no effect in the protein. And a person would not even know they have this mutation since the sequence is exactly the same as the regular protein. The protein will function exactly the same. Okay, but down here now we've substituted, we get a new amino acid. So instead of histidine, we've got glutamine, I believe it is. So the rest of the sequence is the same as the original, but this one amino acid is different. The name for this is called a missense mutation. You don't necessarily need to know that, but it depends on how similar this amino acid is to histidine, whether or not this protein will function properly or it might not function properly. Okay, so take a picture of your work and submit it for today. This is the last question we're going to look at together. It says, state one specific effect on the protein produced if an mRNA code is changed from AGU to AGA. So let's look at what AGU gives us, and then we'll look at what AGA is. So, sorry, I keep scrolling accidentally. Come on. Okay, AGU is right here, and that's the amino acid serine. Now let's look at what AGA gives us. AGA gives me arginine. So this substitution mutation has given me a different amino acid. If you are understanding all of this, is there a way for me to mutate AGU and still get serine? Is there a silent mutation that could give me serine? Hopefully you see here that yes, AGC would also give you serine. So changing that U to a C would be a silent mutation. Okay, so to summarize, point mutations occur at a single point in the DNA. If you look here, if this is my DNA, my mRNA that's transcribed from it would be UAC, and that codes for the amino acid tyrosine. Okay, let's look at 3-point mutations. In this case, an AG has been changed to an A, which gives me UAU instead of UAC. If you look on your wheel, that still gives you the amino acid tyrosine. So this substitution is an example of a silent mutation since there's no effect on the amino acid sequence. Okay, over here I have two other point mutations. If instead of changing the G to an A, it was changed to a C, that would give me UAG. This is a specific mutation called a nonsense mutation. If a stop codon or a mutation causes a stop codon, that's going to have a major effect on the growing protein that's there. And lastly, if the A was changed to a G in the beginning of the triplet, that would give me CAC instead of UAC, and that would give me the amino acid histidine. So instead of tyrosine at that place, I would have histidine, and it could have an effect on the protein. It might not work properly. I hope this helps. Tomorrow, your worksheet is going to be very similar to the practice problems that we did today here, and there will be an answer key posted later on next week. Have a good day.